during this 20 minutes, I will try to a little bit discuss and give maybe some food for thought, and then because there are a lot of things happening, we will dive into 3 examples based on recent research. But so this agenda is, of course, familiar to all of you, and one question is, of course, that what the future also looks like after the emergencies that we have had. If we now try to look forward, when I was a medical student in 1980s, people that were very authoritative in medicine, so they didn't consider a career in infectious diseases very lucrative, as you can see from these comments, that it was rather that we would need to start entertaining ourselves, that there would be something really to do, and I'm not sure that we are so much better today than what the previous generation was. However, what has happened before, so it can apparently happen, and in addition, of course, we have new technologies that can make things better or worse. We of course know that it's not just the pandemics; it's all kinds of outbreaks that can be locally devastating, and we are all aware of the megatrends which bring them to our doorsteps and can initiate new outbreaks. But indeed, the modern diagnostic methods helps to give names to things that just happened somewhere before, and now we know of them. But anyway, if we think about viruses, and I'm a virologist, there are other threats as well. So still only a fraction of them is known. However, just recently, during the last 1 or 2 years, we see papers describing like 10,000 or 100,000 new viruses that are just mined from existing data, and of course people going to the field as well. So we are one species, we have like 20 species around us domesticated, and then there are like 2,000 rodent species, 1,000 bat species that all have their viruses. And just, we of course need the introduction from over the species barrier, or maybe to another continent like Zika virus, and then we need the suitable conditions that allow it to spread, and then lack of control measures just because for whatever reason, but these are not like on-off, but they have a gradient, and the sum of if something is spreading is in a way coming from all these different factors. Of course, basically any virus could, and they are jumping species barriers. This is the Facebook of viruses. So we have some like 30 virus families. They all look the same within the family, but then you have genera within the families. Then within the genera you have individual species, and humans have like maybe 200 viruses in this collection. Not all the families infect humans, and then multiplied with the amount of species, then you have a eternal collection of these. And of course, especially influenza viruses by resuscitations and coronaviruses through recombinations are rapidly changing, and other viruses are doing that as well. So basically, these are the main, among the main groups that are jumping species. Just as an example, so what the pandemics look like, we're still living in a HIV-1 pandemic, but we have had several introductions to mankind, but only then some of them hit suitable conditions and caused a pandemic with 33 million people about now dead. But then some other introductions just stopped there, and maybe a village in Cameroon was infected, and it didn't proceed from that. But there are always more introductions usually than there is space for spread. Influenza A virus, people are very well prepared for, and of course there are the certain conditions where this species jumps are, and if you go to stay here for a while longer and go to hunt in August, and so around if sampled your mallards, so about 10% of them carry influenza A virus. It's all around, but only sometimes then these jumps happen, and you may have the reassortance, and this we know that this has been happening, and now we're looking at the avian flu outbreak with some worry, but we have stuff to fight it, and the surveillance. And then there are like weird exceptions that happen. Now we have a little bit forgotten, but indeed, even a virologist can admit, maybe also prions can occasionally cause devastating infections, and now we know that anyway caused a lot of trouble also this, which was based on just that in the production chain of animal feed there was a change that allowed this to be maintained. There was cow feeding on cows before, but then something happened in the production chain, like temperatures and that caused this prion to survive. But I will now dive to three infections, maybe more. SARS-CoV-2, you know very well. Just to pin out some points: human and monkeypox virus, and then tick-borne influenza. So this is 30 kb RNA. This is 200 huge genome of 200 kilobases of DNA, and then the TB is like 10K. But let's start with SARS-CoV-2, and this is a map of Wuhan. And in December 19, then where the cases then happened of the new infection were centered around the animal market, which is a bit hard to explain otherwise than they were starting from there, and also the not though totally convincing the A and B sublineages starting from there are compatible with the origin there. Of course, the virus has its counterparts in the horseshoe bats, and seems then to be a recombinant that then carried the furin cleavage, when this allowed also the virus bind to an neuropilin receptor and altogether be much more infectious than its cousin, and of course therefore more transmissible. And now we have after that 15 million or so full-length genomes. It's actually much more maybe than is needed. Somewhere it's too little, and somewhere too much maybe. I don't know. But we see that all the old strains before Omicron, they like started from the same point, and then Omicron going then like stepwise forward to escape then the immunity. But if we look like, for example, how to then use the sequence, and we focus on Finland, just we see that whatever happened, or you think of restrictions, so here one sees that the cumulative number of introductions then pretty much stopped after the restrictions started. But anyway, the interesting thing is that it was mainly these countries, namely Austria, Spain, and Italy that contributed then. And if we look at in early 2020, the introductions were mainly from these three countries, and then descendants that entered. So it happened so that individual strains from Spain covered like 1/3 of the epidemic, and 2/3 of the whole early epidemic was seeded by just 3 strains or 4 strains from these countries, showing that actually then it is the much of the dispersal, only some strains, and also the alpha which was coming, we can see that only some, this is the amount of, these are what is being detected at the border, and these are the strains that, and how many strains were seen in the society. Only some go through and start transmission chains, and indeed it is the common dispersal phenomenon. The same phenomenon was seen with future waves as well. For monkeypox, we know that it made it 20 years ago a jump from West Africa to the pet trade in US, and now with also cases noted there, it made the jump to human chains, which then caused the outbreak. If we, a weird thing is happening that there are certain mutations that only occur in humans, which are caused by the human ApoBec enzyme. And with this, you can count when this has jumped the species from whatever reservoir it is to humans. And you can go to 2016, and there it looks like at this year it jumped and has been like going undetected until it entered the super spreading events that caused then this current outbreak. We were, of course, lucky because it didn't transmit better. It might have done that, but it also, this hits I mean, it causes the innate immunity to react better, so it's also less spread, and so there are, you can also look at this in individual patients, and in the same patient you can find versions that have this mutations more or less, so it's an ongoing evolution, and then we can see, of course, that mostly they are imported, and then you have individual clusters, of course, which can be born outside or then within the country. So then the last example is TBV, which is around the forested belt of Eurasia. You have the different forms of the ticks, and you have 2 forms, and they go in both tick species. And here you see larvae, nymphs, and then they are feeding together, which gives them like easier transmission in these rodents. Already 20 years ago in Oxford, it was shown that from outer space, from satellite, you can see where this virus thrives. So it goes around in foci. These foci can be then transmitted by big mammals or birds, and so it's emerging and going up in the mountains in Central Europe. There are more cases, and here's the especially the high incidence area. Now, if we look at the genome of this, and this is the work we recently did in a EVD LabNet project, here you have all these Zikas and Dengues, so it's the tick-borne encephalitis. So we have the European and Siberian subtype in Europe, and if we focus on the European subtype, these are country colors here, so we immediately see that there's not really geographical clustering. It's all over, and when we, so mainly it's the European subtype, some Siberian subtype here, beware. But anyway, it looks like it has come from these sheep-like tick cycle viruses, these 2 subtypes. And weirdly enough, these strains seem to have been born like 700 years ago. If you know better what happened in the 13th century or so, please tell me. And then they have been not really changing so much. But if we look at the general picture in Europe, the strains in Finland are all over, the strains from Germany are all over, the strains from Slovenia are all over, so there is no geographical clustering. This diversity was born in the 14th century. And this is Helsinki, where you are maybe around here. So we have TBV foci all over. Don't worry, it's not enough to be worrisome unless you go to some weird bushes in certain places. But if we look at their genetics, so they are separate. Some is friend with Denmark, some with Estonia, some with Denmark, and some with Slovenia. So they're apart, although there's, again, and it looks from the, that they were born like 40 years ago, and apparently brought by birds, which carry ticks, which may carry TBV. So again, you came to the same, you need the introduction, but also suitable conditions. They may exist somewhere, but you need the bird to bring the tick with the virus. And then maybe the control means, of course, in certain places whether there's the vaccine. So same pattern, of course, this is not going around. So just to control, you of course know the pandemics and epidemics, and these were just some examples. You can eat your vegan meals or something like this to influence the megatrends. You can map viruses from animal reservoirs, and I guess this is pretty much the preparedness and public health in enhancing what has been going on here. As an example, this number 2, what we have done in Kenya, we have screened bats, and then with modern technology and all kinds of biofilm pipelines, we found a new Ebola virus. Just the same time, the Americans found it also in the other part of Africa, and so it's still out there, but doesn't seem to maybe be that pathogenic. But however, it looks like bats running around. They have Ebola virus infected cells in their lungs, but the lungs look otherwise okay, so no pathology, and this is what you would expect from a reservoir. So it looks indeed that these species are a reservoir of the Ebola virus. So in general, there are of course all kinds of projects in EU mushrooming around this, and VEO, Virtual Emergency Observatory, led by Marion Koopmans in Erasmus, is one of them. And the one idea is, of course, that for One Health, you're going like from the spillover events before you are seeing something in man, and try to see what is happening here before this outbreak starts. And if we ask Anthony Fauci, this is what he wrote in 2020. As human societies grow in size and complexity, we create an endless variety of opportunities for infectious agents, which will bring us life-saving drugs, vaccines, and diagnostics. But anyway, we can expect ever more frequent and deadly emergencies of infectious diseases. So with that, this is just the common theme from Marion, that instead of looking at this from an anthropogenic angle, so we should see that humans are just one species, and these are the people that contributed to the TBV study, and there are countless colleagues that have contributed here. Thank you.